Welcome to Life Science Solutions. I'm your host, Chris Adkins, and today's episode is a special live recording with Ryan Doxey, Vice President of Technical Solutions at Kymanox, where we have part one of a conversation on hyper-concentrated formulations and their benefits, including hurdles like viscosity, stability, and commercial scale-up. From sweet tea analogies to hard-earned lessons, it's a great chat about what makes these technologies both technically challenging and clinically promising. I'm really excited to be here today with one of our own team members at Kymanox, Ryan Doxey. So Ryan, welcome.
Well, thank you, Chris. It's a pleasure to be here, and really excited to be live on this one. This is a first for me. So
I know.
Really
It's a twist. Like, I feel like I'm living out my childhood dream of being a news anchor.
That's right.
Right.
Yeah.
So it's pretty fantastic. Well, what we're going to touch on today is we actually have a panel coming up. There's a pretty well-known industry conference in the fall every year up in Boston called Partnership Opportunities in Drug Delivery. Sure. And where you're actually participating in a panel that is all about delivering these hyper-concentrated drug formulations.
That's right. Yeah.
So that's a doozy of a mouthful. Hyper-concentrated is one of those fun words. You don't normally get to say it in your day-to-day, right? So can you talk a little bit just about maybe first, what kind of falls under your remit at Kymanox, right? And then second, what is a hyper-concentrated formulation and why do we see so much talk about it in the industry right now?
Certainly. So within Kymanox, my remit is I lead the technical services group, which is for lack of a better term, the CMC, drug product constituent development portion of a combination product. Mm-hmm. So we develop both the drug substance and the drug products, and prepare them and get them ready to go into their primary container. Select the primary container that's most suitable for that product, and then if there is a drug delivery aspect of that, hand it over to the combination product group to then develop it as the combination product with the delivery device. So that can be an auto injector, pre-filled syringe, on-body wearable, nasal device, inhalation, any delivery device that gets the drug into the body. So
because the drug does you no good until you can actually take it, right?
That's correct. Yep. So it's the advancement beyond the typical pill or single injection with a syringe and vial. So the more sophisticated at-home use for injections. And so the hyper-concentrated formulation technology is really an up-and-coming game changer, in my opinion, specifically with the monoclonal antibodies. So, monoclonal antibodies, proteins, once the concentration starts getting, that 150 to 200 milligram per mil, you start seeing aggregates, and, the protein doesn't like playing with itself. It begins to crash out of solution. So these hyper-concentrated technologies, what they do is they really dehydrate and stabilize the protein or the monoclonal antibody into a form to where you can pack a lot of it into the formulation without the interactions. So you stabilize it, and you're able to get 400, 500, sometimes 600 milligram per mil. So twice as much as you traditionally would. So it's a huge game changer in the field of monoclonal delivery.
Yeah, I think one of the ways that I think about it, being a native Southerner, growing up, we made sweet tea.
That's right.
And everyone who's worth their salt in the South knows that you start with your hot tea. You put your sugar in it and you put a ton of sugar because that's how you make it, right? Then you cool it off.
Yes.
Because you can't do it in the other order because you won't actually get the sugar to go into solution. As a kid, you'll kind of learn, you'll get the cup of tea at a restaurant and it's unsweet. And you're like, well, I'm going to put some sugar in this. And you realize that the fake sugar stuff solubilizes, goes into solution different than the real stuff, right? So that's kind of the layperson's exposure to this. But in drug development, I mean, this is a really big deal.
It is a really big deal.
Right.
Yeah. So, the other alternative is the high injection volume. So getting more of the antibody in the subcutaneous space by using an enzyme like hyaluronidase, right? Which removes the superficial or the subcutaneous tissue to create a void to allow more injection volume. So you don't have a loose pancake, floating on top of, right below the skin.
Yeah.
So, with the with the high injection volume, translate that into an auto injector. I mean, you're looking at a horse leg level injection, an auto injector that you hold, with two hands to inject. Patients don't want that. They want, their, 1 to 2 milliliters under the skin, get your medicine and go about your day, rather than having to wait and do a long 30-minute injection time. I mean, no way. I mean, 30 seconds is long enough.
Even 45 seconds or a minute. No one wants to hold that, horse leg, table leg, chair leg, whatever analogy you want to use, right? No one wants to hold that against their leg for that long, because whenever we say it like this, it doesn't actually feel like that long.
That's right.
And then you ask someone to, stop everything that you're doing and concentrate on doing something.
Yeah.
And let alone if you have a patient population that there's any kind of dexterity issue, right? Or you've got any kind of, mental acuity problems or anything like that going on, So maybe let's give some perspective, because this is we're pretty niche, right? We're already niche in life sciences, but we're getting niche here. Everyone has gotten exposed to auto injectors now, right? The rise of GLP-1s has really put it out there. They've been around for a while now in a relative sense, but what's a typical delivery volume in your standard run-of-the-mill auto injector that maybe someone's getting from, Lilly or Novo Nordisk?
Yeah, so they range from 0.5 ml to, 2 milliliters. And, it's all based off also on rate of injection. So, viscosity plays a key point, and we'll touch on this a little bit later when we talk about, the high concentration formulations, but, the viscosity of the solution, and the injection duration the injection rate plays a lot into all of this. So, it's volume over time. And the faster that you can get it in, I mean, you think of the EpiPen, right? It's, millisecond delivery, instantaneous. Where some of the larger volumes, depending on that viscosity, you're going to have to hold it there for 10 seconds, which is an eternity when you're holding a needle under your skin.
Correct.
Correct. So,
right. And heaven forbid you get into any kind of like fear of needles or,
right.
Hypersensitivity to the pain and things like that. Right. And EpiPen gets used in an emergency situation. It's a needle big enough to go through our jeans,
That's right.
So there's some different considerations there.
And you think about, the EpiPen example and the emergency use, when you have a chronic disease that requires, repeat dosing, you're searching for new spots to deliver it, because you do get sensitive, areas and injection site reactions and things like that. So, to ease the burden there, for the patient populations, I think it's critical. And this hyper concentrated formulation technologies is really an advancement in that, and really the key way to get it. And also, I think one of the most underrated, important aspects of it is, the potential to co-formulate antibodies. So, because you've dehydrated and kind of increased the stability of this antibody, you can put 2, maybe 3 antibodies together, to address different targets. So, there's a lot of opportunity in this space.
And something that, you see if you start looking around, I feel like I see it, especially in the oncology space, a ton of co-delivery, right? Like, we've marketed this drug to be an add-on. I'm sure there's a very sophisticated clinical term that as a manufacturing guy, I don't know. But we've made this to go with, whether it's Keytruda's pairing with a dozen different things for different cancer indications, right? So potentially there's a better way of doing this.
Right? So multiple injections. So now you have, the burden of finding a spot for multiple injections that aren't IV, of course. So anything intramuscular or subcutaneous is the kind of the topic that we're speaking here. And that's the in-home use, right? You can't do an IV at home. I mean, let's just face that.
Unless you have a full-time care person.
That's right. That's right. And a PICC line, so, so, trying to find all of these different ways to it's really easing that patient burden. I was thinking you brought up a good point around this co-administration, and, that really being the advancement of the future. And I really see that for possibly lots of kind of orthogonal indications as well. I mean, can you capture, a lot of the autoimmune type diseases with a single shot, and then around the personalized medicine aspect of it as well. So, there's lots of opportunity, and, we still have a ways to go, but it's in sight.
Yeah. I want to lean in for a second on that patient burden piece, because this is something that if you're not in the demographic that has to deal with these issues, right? Like we can just kind of completely gloss over, but there's instances of, whether it's in different cancer regimens or you hit on autoimmune, lots of other conditions that require, maybe repeat injections over weeks or over months. And for plenty of these medications right now, the standard of care is you go to the hospital every week.
That's right. Yeah.
Right?
So clinical visits, healthcare provider, administration,
which has a cost impact too, right?
That's right.
But even beyond that, like, I mean, let's really hone both of those examples, right? Both cancer and kind of anyone with any autoimmune, especially if they're to the point they're taking an immune suppressant. The last place in the world they should be spending any time is a hospital.
That's right.
Right.
That's right. Yeah.
I mean, we could clean them every minute, and they're still just like might as well be petri dishes of disease. Every third time I go to a hospital, I get sick, I think. Right.
yeah, for sure.
And so, but getting these therapies to be home, presentable is a real challenge.
It is.
Right. And we see developments all over the place. I mean, this is kind of, I would argue, the highest science version of innovation that's going on here. We have other things like even the prefilled syringe, which in pharma world is actually still pretty new to be a standard delivery mechanism. We continue to see it getting iterated, right? So now we see, ultra thin wall needle cannulas that are just to reduce, to your point, the injection force so that injections can happen faster. They're not really enabling, maybe they're enabling slightly higher concentration, but they are more comfortable.
They are.
Right.
Yeah.
It's less skin that you're messing up to get the same amount of volume flow.
That's right. That's right. Yeah. And so, all of those aspects play a really key part. So, I'm going to take you back, 20 years ago, and we talk about the patient burden and so back then it was the pill burden, right? So we've been here before in this industry, even before auto injectors and prefilled syringes were, common practice in the industry. We were talking about pill burdens and how many pills does a patient have to take. So, trying to reduce that pill burden, 10 to 20 years ago was at the highest, front of mind for almost every oral company. And, moving into the extended release, modified release, to try to reduce that pill burden. So we've been there. We know how to overcome it. Yeah. It's just a matter of how we do that. So, the formulation technologies are really evolving with the administration technologies. So, that's kind of the point I was trying to make. We've seen it with the oral. Now we're in it with the parenteral, sub-Q.
And there's an overall benefit here too. I mean, we lead on the patient side because that's where our brains go first, right? But there's an adherence benefit, right? I mean, trying to actually take a pill every 4 hours or 6 hours, every 4 hours is miserable, right? Last time I checked, I like to sleep more than 4 hours at a spurt, right? Yeah. But even every 8 hours is actually challenging to remember.
It is.
Right? It's a weird cadence to keep, kind of in perpetuity. Talk to anyone who's on any kind of pump or patch-based system for Parkinson's right now, right? They're all on medication that's in and out of their system, and they're measuring up and down time. And a good day is, 6-7 hours of uptime. So it does have a real impact.
Yeah. It's funny you mentioned that. So I'm actually dealing with that at home right now. So my daughter's on an antibiotic, and 3 times a day. And my wife and she was trying to calculate it out perfectly, such that she gets 8 hours of sleep and still gets it 3 times a day. And I'm like, honey, that just doesn't work.
You got to anchor it to something, right? It's got to be breakfast, lunch, and dinner. And yes, I know that means breakfast and lunch are closer, right? If you're a typical person, right? But you got to anchor it to something or else, and this is one of the actual really interesting differentiators at Kymanox, because we don't only work in the technical and the delivery. That would be enough to be unique, right? But we have a whole human factors usability team, right? To make considerations like that. Right? What is actually the best frequency that we're going to get good adherence and patients are going to understand it? And then how do we go test that, right? In a formative, and then prove it in a summative. Right. So that's a good kind of cross-platform perspective to bring.
Of course.
So I think that paints the picture really well. And to really, we want to talk about, kind of tease a little bit for this upcoming panel at I'm never going to call it Partnership Opportunities in Drug Delivery again. It's POD, as everyone in the industry knows it as. And, the purpose of this is to bring together, yourself, some thought leaders from both big pharma as well as the innovator side in this space, which we see a tremendous amount of innovation going on, right? And some of it here local in RTP, but certainly all across the US, right? And so, can you talk a little bit about, maybe briefly from the technical side, right? What makes this so challenging as we get into the higher concentrations, and why can't we just do our standard methods of higher concentration, right? And maybe touch on precipitation and some of the other issues that tend to happen.
So the key challenges with this technology is, as I mentioned early on in the conversation around the dehydration and, the stabilization of these dehydrated monoclonal antibodies. So you can't put it back into an aqueous delivery vehicle because it's just going to rehydrate, right? So you have to do the manufacturing processes to generate these have to be non-aqueous in nature. So, a triglyceride based delivery vehicle, something non-aqueous, but can also, be absorbed by the body, right? So you can't just pick something off the shelf and inject it in humans. It has to be, safe and deliver these antibodies, in a safe and stable manner, right? So, the solvent exchanges involved and the collection of the dehydrated material is really key. And so it has to be done aseptically. So, free of any microbial bioburden at all, right? So sterile. And that becomes a challenge at a larger scale, right? So, we can do this all day at the bench scale. We can do it for animal studies. But the real challenge is getting material that is sterile for for human use and clinical use. So, that's the real burden right now that these technology companies are facing. And there are solutions out there. It's a time and money, exercise and trying to get it without, such a financial burden to the company.
Correct. Right. Because, we both come from kind of manufacturing or manufacturing adjacent backgrounds, and really keeping in mind, right? Like how do we not just make 5 or 10 of something, right? Or in this case, right, how do we not just make 50 or 100 or 150 doses, right? We've got to be able to make, for some of these products, right, millions of doses, depending on the specific indication. And it does us no good to do less than that. So when I was in primary packaging, we had this conversation all the time. We could make the most bulletproof, coated elastomer that you could package and it would not let anything in, and we could make 5,000 of them a year, which would do nobody any good. That's right. So I don't know, maybe some gene therapy companies, but no one else. So it wouldn't even make sense for us, and the economics do come into play, right? And we see tremendous conversations right now. We won't talk about this, but simultaneously about bringing drug production back, but also lowering prices, which it doesn't quite mesh. But then also in advanced therapies, and how do we bring the pricing down?
That's right.
Right. And whether that's, the GLP-1s, it gets more heat because of the big price tag in some of these, cell and gene therapies that are more curative in nature. But it does no good to come up with the technology if we don't have a way that we can reasonably monetize it.
It has to be industrially feasible, right? And it has to be able to supply the market need. I mean, the products that you're talking about here, with these monoclonal antibodies are big, big volume, highly prescribed drugs.
A lot of even lay people are familiar with Humira, right?
Yes,
for
Humira,
years was the number one selling.
Yes.
Skyrizi, Dupixent, right? I mean, ads are all over TV. I mean, you can't watch a football game and not get 4 ads for all of those, and then some GLP-1s thrown in there,
for sure.
Injectable, small molecule, but still injectable. So, what are you hoping that you guys will get into at the panel, right? I mean, so it's unique to have, and not only do you have pharma, but you've got, kind of innovator side, you've got generic side, you got a moderator from AstraZeneca, you've got Teva on the panel with you. You got yourself from kind of an external, perspective. And then you've got one of these innovators that's kind of bringing forth this enabling technology, right? So that's a unique culmination.
It's a great culmination, actually. So you get all 3 perspectives. So you have the large pharma need with, the the drug substance, the drug in mind. You know, they're trying to look at this as a life cycle management play, right? They probably have a once weekly dosing, you know, product that they're trying to get to once a month, or, you know, once every 6 months. You have the innovator over here who's trying to develop the technology and demonstrate the value, right? And it's a play between, you know, how quickly can you do it and how quickly can we get it done?
Yeah.
Right.
Yeah.
And, you know, my perspective is kind of the middleman here. How can we best get it done such that it meets the needs for both companies, right?
Yep.
And, you know, I think we're in a unique position at Kymanox because we are able to see, you know, the field of view. We see the forest through the trees. Where a lot of companies get so honed in on their specific application that they lose the focus of the entire forest. So, you know, with this panel, I'm hoping that we can really start to see the forest through the trees, and initiate conversations on, you know, how to best meet the patient need. And, you know, at the end of the day, it's about the patient need.
Yeah, absolutely. You said something in there that I want to hone in on some. So you see it as a life cycle play, right?
Yes.
So that would, like you said, a drug that's on the market that right now has a higher injection frequency than what would maybe be ideal, right? So my impression is generally these technologies, I mean, you've commented on this, they are significantly formulation impacting, right? And so I would expect pretty significant clinical burden on making the change, right? To confirm bioavailability, that PK performs the way that you would expect it to, as you're already adjusting dosage frequencies. You're already going to confirm that, right? Do you see anyone, or do you maybe project that anyone might be interested in it for a novel therapeutic that right now, either because of just what it's going to take to get dosage efficacy, right, or what it might be, that they're just going to need to inject so much that maybe this is the only way they can get any kind of reasonable concentration?
It's possible. Yes. I think
I know why you don't think anyone, but I'm curious to get there.
So, you know, here, so we tell our clients, I tell my clients this quite often, you know, so if you're considering this technology, get in line, because once it's at a point to where they're dosing in the clinic, the line's going to be out the door. Right. So no one, I mean, we're in an industry where to be the first is dangerous. Right. So, you know, people kind of shy away from being the first for a novel technology, but once a novel technology hits, it hits. Right. And so
and then it's join the queue.
It is. And so if this is of interest or of need, you know, get in the queue, because the queue is going to get very long very quickly. You know, I have tremendous amount of confidence in this technology, and, you know, it's really just breaking that barrier to be able to get it into the clinic. You know, on the clinical burden and all that, I think the clinical burden is going to be a little bit less than people initially think. I mean, it's definitely not, you know, a 505(b)(2) where you can just do a comparability protocol and say that they're equal. There's going to be some clinical dosing for sure.
Sure.
But the technology, it's not changing the native antibody. Right. So you're just super concentrating it. So it's a safety thing. And if you can demonstrate safety, and then, you know, bioequivalence, it's, you know, you have to show that 6 month interval is the same efficacy as that 6 1-month intervals. Right.
Correct.
So
that's gonna be where your proof's gonna be. So
that's right.
You know, and it's interesting because normally we talk to folks a lot about different delivery technologies, right? And we talk about everyone in the delivery technology world, everyone's waiting for commercial approval. Then that validates your delivery technology, right? Partially because you frequently don't bring in your final delivery technology until phase 3, sometimes a phase 3B. It just kind of depends, but frequently you're moving your way through the traditional, okay, I'm in a vial because I'm doing variable dosing. Now I'm in maybe a couple of syringes, right? And then I'm going to go into an auto injector and I'm going to bridge in a safety device or something like that. So commercial approval is kind of the marker for their success. So it's interesting here, and I think I could guess why, right? But you comment on clinical approval being the marker, right? And maybe highlight why there would be that difference between these two if you kind of turn your head sideways, maybe similar accomplishing things, right? I mean, an on-body delivery system lets you deliver a higher volume reasonably without using a horse leg and get more drug delivered. And ultimately these do that as well, but they have a different driver in their approval mechanism.
And, for what it's worth, animal studies, when you're talking about biologics, are just that. An animal immune system is not the same as a human immune system. So that translational, the clinical translation is not there like it is for a small molecule. However, the animal data that we're seeing is tremendous, and the safety signals that we're seeing are tremendous for these high concentrated monoclonal antibody formulations versus the native. And we believe that translation into human will really be there, and to get it into the clinic, it's demonstrating that it's a viable option for further development. And so I see the industry really like gravitating to that clinical use and really starting the life cycle management play a lot earlier. And you're able to bring this into the development program later than you would an on-body delivery system or something like that. And so, think about that. That's, another option for, longer injection times is that on-body device. It's a wonderful
and you
achievement.
can deliver 5, 10, 20
That's
mil,
right, over time. But again, who wants, this on their body for so long? So anyway, so
it's interesting because when I think about it as well, I think, okay, the we know that the technologies work, right? We know that we can demonstrate them at the bench, right? But how do we make it in a way that's appropriate to give humans, right? So the aseptic, the particulate piece, right? Because you're gonna end up with some concentrated formulation that you can't just sterile filter, right? Which was like, as an upstream guy, and you got to purificate, you got to the beginning of purification, and we ran through depth filters in open processing, which a day ago would have been unfathomable, right? Because we were going to sit it there in cells and in buffer and let whatever got into it grow for a whole day. But now we're going to sterile filter it, so everything's fine.
That's right.
It's not quite true, but it's roughly true. And so you remove that ability. So when I think about it, I think, okay, well, whoever gets an IND approved with the technology, right? That first one for, whether it's Lindy or Electrify or Xeris or and now I'm forgetting one, Nanoform. Thank you, the one who's on the panel with you. So whoever gets that first one approved, that's going to be of significant review in the IND.
It is.
So it's going to be technology they're not. Well, maybe they're familiar with it through an emerging technology program.
That's just it.
Hopefully they are.
They are.
I would hazard to guess all 4 of those guys have submitted to that program. I would hope they have. So, but that's where that's going to get a lot of scrutiny is how have you done this in a demonstrated aseptic fashion?
It is.
Right.
And I think that's really going to be. It's going to be the key is getting through that, the CMC reviews, and making it congruent to what the regulatory bodies are used to seeing, in terms of, particulate matter and things like that. I mean, you have a suspension, you're going to have particulate matter by name alone, right? So being able to distinguish between the extrinsic particulates and the drug particles, it's going to be a challenge. But again, they're not all challenges that cannot be overcome.
Sure. Right. I mean, we visually inspect all kinds of opaque, looks like cloudy milk essentially, right? And we managed to validate that, right? I mean, I think about what the regulatory expectations are going to be on things like media simulation, right? I mean, making sterile product once isn't good enough for the agency to let you, take care of that, right? So, well, this has been really good, right? I enjoyed this chat. Again, this panel's coming up here in a couple months. We'd love to hear from anyone who has things that they're curious for the panelists to get into hopefully if you've made it this far, you're a hyper concentrated formulation guru, or at least geek, right? And that means you're probably going to be at Pod, and our team would love to chat with you.
Yeah,
then.
definitely. So, if you made it this far, appreciate it. And looking forward to carrying on the conversation. I mean, I think we could probably go another 30 minutes.
We easily could, right? And if you're just here in the podcast marathon and you're just like wandered into Nerdville, then welcome, and we hope you enjoyed your stay. So awesome. Well, Ryan, thanks so much. This has been really fun.
Thank you, Chris. I appreciate it.
Yeah, awesome. Thanks for tuning in to Life Science Solutions, presented by Kymanox and produced by WalkWest. For more insights like this, subscribe and follow Life Science Solutions wherever you get your podcast. We'll see you next time as we continue exploring what it takes to bring life science innovation to life, because patients deserve better.